right, it's Peter here with my FSHD. Probably can't see it in the background, but I am here in Boston at the American Society for Gene and Cell Therapy meeting. It's our annual meeting, and I just gave my presentation on our CRISPR inhibition program. Actually, I was talking at the session on overcoming hurdles to musculoskeletal gene therapy. And I talked about Carissa's Have a Little Heart and our very specific gene expression regulatory cassette that we optimized for FSHD, but it's applicable to a number of different neuromuscular disorders, highly compact and great for AAV-mediated delivery, where you need expression of your transgene in skeletal muscle and just a little bit in heart, or even none in heart. I know it's kind of loud because I'm outside because I got my Dunkin', my large hot regular, and of course I got my chocolate glazed. And so the breakfast of champions when you're in Boston. And anyway, but I'll be going inside where it's quieter and talk to you a little bit. We're going to go over, so, you got to be an idiot to have a podcast if you're a scientist, let me tell you, unless you're Neil deGrasse Tyson, which actually I love his videos and stuff. They're actually pretty cool. But other than that, I'm no Neil deGrasse Tyson. It's just, you go fast, you try to simplify stuff, and there's always somebody who points out, well, you can't do math, or there's a typo, or you just didn't really explain this right. Now, you got to remember, come on, guys, the podcast is for you patients out there, you families, you non-scientists out there trying to bring you some of the science. I do appreciate the scientists out there keeping me straight and keeping me online. But sometimes I oversimplify things. I understand. I'm glad that people pointed out to me that there's some confusion because I actually always want to get it right. Even when I simplify it, I want to make sure I get it right. So we're going to go back to my Excel sheets that I did with Brad, which I had whipped up in 5 minutes before getting on the phone with him. And we're going to kind of go over a little bit about how we can look at gene expression data because, the Avidity trials come out, the Sarepta Arrowhead trial. You're going to be getting other trials coming out. At some point, I assume CRISPR is going to be showing their expression data. Souffle will be showing theirs. There's a lot. And, I'm going to show you what I look for in the data, how I would present it, how they are presenting it, how it's different from mouse data, and we're just going to cover a little bit of that. After that, I'm going to go ahead and I'm going to give you my pig talk, because that was the second part of my talk on overcoming barriers in neuromuscular disease, was large animal models in neuromuscular disease, and why the pig is so important, and how the pig actually helped us. So anyway, I better go inside because it's really freaking loud out here.
All right, well, as you can see, I am not in the convention center. It turns out that 20,000 other people had the same idea. Hey, let's just it's a big, big space. But it is really hard to find any place that's private once you go in there. At these large meetings, if you pay to attend, you get this little QR code, and then you can live stream any of the sessions. And they have maybe 4 sessions going on at the same time, and with 4 talks in each one on different things. And if you want to see talk 1 here and talk 2 over there and talk 3, it actually hinders you to go to the session. So people do is they find a table, they find a chair, they find a little corner somewhere, and they're just watching the sessions they're interested in. And you can catch them all. You may be able to bring multiple ones up. I don't know. Anyway, there's no space or privacy anywhere to run to do the podcast. I just felt like I'd be bothering people talking. Most people are actually pretty polite about it. So anyway, so I took the time instead to put together some information for you. I'll do a little bit better job than I did when I was on with Brad to cover how I prefer to interpret gene expression profiles from knockdowns in these clinical trials like the Avidity and the Arrowhead Sarepta trials and the ones that will be coming up, the previous Fulcrum trial. So we're going to talk a bit about MRI and stuff. But briefly, I can give you a recap on the meeting. It was the American Society of Gene and Cell Therapy, and in regards to neuromuscular disease, or anything relevant to FSHD, there was very little, truthfully. David Mack, from the University of Washington presented on their 3D culture system, but quite honestly, they're like years behind Silver van der Merel's 3D muscle system in Leiden, which already has FAPS incorporated into it, fibro progenitor cells, FAPS. You know me, I'm always horrible with names on things. And starting to add in other things, but essentially 3D muscle culture for, what's relevant to us is FSHD, which is interesting for some things, not so interesting for other things. After I talked, we had Melissa Spencer from UCLA talking about the immune response to AAV gene therapy and DMD and what they're learning from those trials. So, it's pretty cool. And what it basically says is there's a lot of individualized immune response to AAV, especially high dose AAV. So that's again a high dose AAV. When you use something like RH74 or AAV9, just it's a different game than using these new myotropic vectors. And then Amy Wagers from Harvard talked about some base editing CRISPR targeting that they're doing to kind of change the sequence of genes, which quite honestly is still pretty inefficient, but put a good spin on it. But it's cool technology. She's always at the leading edge of genome engineering for neuromuscular disease and stuff, and for DMD, mainly for DMD, but also they do some other diseases. So that was pretty cool in our session. But there wasn't a whole lot else. Scott Harper's lab had some posters on their typical programs, FSHD, but I think the big thing that was interesting to me. So, after I gave my talk, because we have not platform, we have a technology, a cargo for gene therapy, right? And there aren't that many, right? Modalis was there with their epigenetic silencing, which is just a knockoff on what we're doing. CRISPRi had some posters, but they weren't talking about FSHD at all, which is quite surprising because if you had good data, you'd think you'd be trumpeting it. No, where was I going? I can't remember. Oh, that's right. So after I gave my talk, people, coming up, everybody and their uncle has the new best-in-class myotropic AAV that's going to be great for skeletal muscle, actually striated muscle. Everyone gets into skeletal and heart, and way better than myo AAV, way better than AAV myo. AAV9, RH74 are distant. No one would use those anymore. But it's just amazing. I mean, the technology, the AAV, just when you think, boy, at some point you got something as myotropic as you can and liver detargeted as you can, and yet they just keep coming up with the next generation, whatever. And so there was actually some really cool technology, a company called Dino, which I had never heard of, has a whole series on these things. And they're basically just a virus company that want to make virus for your disease. You give them the cargo, and work together, and it goes to clinic. So, and there are a number of others that actually had their own everyone's best in class. I guess no one's going to say mine's the mine's the second best in class. It's all spin, and that's one of the things we're going to talk about is how to spin, how people are spinning stuff. But yeah, there wasn't a whole lot there. I got to tell you, not a whole lot for neuromuscular disease. A lot of vendors, very big on CAR T therapy and these types of things. But anyway. All right, I got to meet my friend Chris Rogers, CEO of Exemplar Genetics, where they have the pig. That was actually cool, and we drummed up a bunch of interest in the pig. I think for not just FSHD, but as a model for fibrosis and NMD pathology that needs to be addressed. Because I told the audience, I said, like, all of y'all are going to, treat your neuromuscular disease of interest, and you're basically creating another neuromuscular disease. Somebody who had SMA or somebody who had DMD or LGMD or FSHD, and now that the pathology is not being driven by the primary lesion, genetic lesion, for example, by DUX4 anymore, or by loss of dystrophin or by loss of SMN, but you have all this pathology you need to get rid of, and ways that we can accelerate this so y'all can get your muscle back, get your functionality back. That's why I think the pig would be applicable to a lot of people. But we'll talk about the pig. I'll talk about that a little bit after the break, and so I got to do my pig podcast, my pig cast coming up. All right, but before we get there, I just want to go back over briefly my comments about the Avidity trial and the Sarepta Arrowhead trial. And I've come out and said that My new favorite is the arrowhead approach. And I've actually, of course, I get some grief. Turns out, well, God knows, you never know who's listening to a podcast. It's out there for everybody. But the thing to remember is because it's out there for everybody, and I can't hide from what I say, much like many others who say things quietly in private. I, I'm putting my name behind it. I'm putting and I gotta tell you why. It's putting myself out there, opening myself up to ridicule and risk and whatever. So let me tell you. Okay, first off, siRNA, small interfering RNAs, little double-strand RNAs, they work. Okay, antisense oligos, they work as well. Maybe not as good. They work through different mechanisms, but in the end, knocking down DUX4 mRNA. FSHD is the ideal disease for antisense technology. Okay, siRNAs, ASOs, short hairpin RNAs driven by in gene therapy, right? You'll hear about these things coming. That's what the Kate Novartis is doing, the Harper lab with Armatus Bio, right? It's all the same. It works really well. Okay, knocking down DUX4 and should have very little off targets. So what's the difference? Okay, why pick one or the other? Okay, it's actually, in my mind, not about the efficiency and the efficacy of the knockdown. They're all really basically going to work. Does it matter if one works 70%, another one works 80%, and another one works 90%? To me, it doesn't. That's not what's driving my decision. Even 50%. That's actually not what gets me excited about things or depressed about things. What matters to me, number one, is going to be durability. How long does the effect last after the injection? Okay, number two, it really should be number 1A, 1B. I guess that'd be 1A, 1B. Targeting. How are you getting it into the muscle? So why do these two things matter? Well, durability is how often you're going to have to go to the clinic, which is going to relate to cost, but also, you're going to have to get yourself to the clinic, and you think about yourself, oh, I can get to the clinic, no problem. But can the clinics take you? Can the clinics handle you? The volume of people, because it's not just FSHD. It's going to be DMD, it's going to be SMA and LG. I mean, just huge volume, okay? An aspect of that is going to be, is it an IV infusion and how long does it take? Or will it become a sub-Q? Right now we don't have any subcutaneous delivery mechanisms, but that may get there and help alleviate the problem. But again, you have to go every 6 weeks, every 12 weeks, twice a year. That's going to really matter. I would, if you had a therapeutic, an siRNA that was delivered twice a year that showed, say, 60% knockdown, that was durable for 6 months, versus, say, an siRNA that had 75% knockdown, but you had to go in every 6 weeks, I'll take the one every 6 months, because you know what? That 60% knockdown is going to be good enough. You're going to get therapeutic benefit, tremendous therapeutic benefit. Okay? So durability to me is matter, but also, okay, so then mode of infusion, sub-Q versus IV. We don't have anyone doing sub-Q, although I believe everybody's trying to get sub-Q, so they'll be coming. But right now, let's say everybody's IV. Then how do you get targeting? So avidity is targeting the transferrin receptor. Why does that matter? Well, that doesn't turn over very fast. It's on all the cells. People say it's enhanced muscle delivery. It's not really. It just gets it into muscle, but it gets it into everything. In fact, the reason you use transferrin often is to get into the central nervous system, actually. It's for other diseases, which you don't need to do for. But it's not like just getting into muscle. It's getting into everything, right? But the problem is, this is where you get some toxicity with dosage. You can overload the system and you have adverse effects, which is why they had to lower their dose. And long term, we'll see. And, if people are going to have adverse effects from chronic blockage of their transferrin receptor, that's one possibility, right? So what about the arrow going into the alpha 5 beta 7, I believe? This is different. This actually recycles very fast. Again, this isn't just into muscle, it's getting into other cells as well, okay? You only really need it into your skeletal muscle, but it recycles very fast. And what they've shown is that, so if if the cell, your body, needs to use, it's going to be available. It's not all bound up with these targeting moieties that are getting the siRNA into the skeletal muscle. And the second thing, so they can actually increase the dose. So if it only works at 60%, they can actually crank it up, go higher, try to get to 100%. But do you need to get to 100%? I'd argue you don't need to. everyone's walking around with a little bit of DUX4. Asymptomatic people have a pretty decent amount of DUX4. Okay. So, well, but it doesn't matter. You have that opportunity. And I predict there will be less toxicities, if any, associated with it. At some point, everything's toxic, right? Water's toxic if you go high enough dose. Everything, right? So, okay, so that's the delivery. The durability. The avidity had to, they're at every 6 weeks, as I understand it. And the arrowhead, people, oh, but they had to drop from 12 weeks to 10 weeks. Still a lot better than 6 weeks. Okay? I mean, and again, I got no stake in the game. I don't work with any of these companies. I know people at these companies just because everybody uses our mouse models and stuff and things, but I have no relationship with these guys. No stake in the game, nothing to gain or lose with any of these. I'm not cheerleading anybody. I'm just telling you how it is. This is why I like it, okay? It's not because people came and said, well, can you really trust their level of knockdown for the Arrowhead put out? They're missing samples. Yeah, I know. And how you compare it. I actually don't care. We're going to go over some data. It works. And, what I want to see is durability and the targeting. Then the next thing is going to be, it's going to come down to manufacturing, which is going to get to cost. The siRNAs have a cost to make those. Then however you link them, whether it's an antibody fragment, or a peptide, that actually is expensive. And then the linkage is inefficient and even more expensive. Okay, and this is going to get to cost to you. Okay, so how often do you have to go and how much is it going to cost? That's really a lot of how much it costs to make it is going to be a big deal when it comes down to manufacturing and the efficiency. Okay, so these are the things that matter. Does it matter if this is 82% versus 85% versus 96% versus 78%? It might matter to investors. It might matter to, people want to say we have the best knockdown ever. But, the other thing about that is you really can't compare their knockdowns because the way the data is reported, and we're going to talk about that, right? Did you use MRI-informed, non-MRI-informed? How, it's not so much what genes you're looking at, but how you do the math, what you do the comparisons to. I think that actually companies don't necessarily want there to be able to be a direct comparison, right? What matters to me, and the reason I said I like the Arrowhead Sarepta, I should just call it Sarepta, I guess, just because people, it's siRNA approach is durability and targeting. Probably still pretty expensive to make, but right now, that's what I like. Others that are coming, that's what I'm going to be looking for. Durability of the effect, how they're being targeted, okay? And ideally, I would love to see sub-Q delivery instead of IV, but, I'm sure that's coming. But these are the things, okay? And so those of you, as I said, so when Avidity said they have between 23 and 78% knockdown or whatever, and Sarepta showed that they had like up to 90 some percent, they both work. That's what I got out of it. That's actually, what I want to see is they work in everybody. Everybody goes down. We're going to cover that. Durability, okay? And how are you targeting? Because that's going to affect potential adverse events and toxicities, okay? How is it getting into the muscles? Those are the things to look at. It works. You optimize them like crazy, but they pretty much work, okay? The technology works, okay? All right, so let's go. We're going to talk a little bit about MRI and then gene expression. I'm going to take you through some of the data actually, because there's some real, we just throw it out there and the amount of work and the people involved, really, astounding. I want to make sure credit where credit's due. Most of this work came out of the Seattle Wellstone and the Leo Wang, Seth Friedman, Stephen Tapscott, and those gangs up in Seattle doing fantastic work. A lot of it funded by Friends of FSH Research as well, especially the early work before NIH got involved on MRI and the gene signatures and the impact of this stuff, because we all take it for granted right now. DUX4-induced targets. Let me tell you, there was a time not that long ago when people were still questioning whether or not DUX4 was the target. So let's go to the screen share. All right. So I know you guys know what MRI is, or do you? I don't know. So just remember that's magnetic resonance imaging. So it measures how water molecules respond to magnetic fields and radio waves. Okay. Let's see if I can't get a laser pointer here. Okay, so normally if you've gone to the doctor and gotten an MRI, you're gonna be, these things right there, gray, white, black, and they look at your different tissues, right? And typically your medical doctor is gonna focus on hyperintensity regions, right? Because these are regions where there's something wrong, something abnormal, right? And they're usually bright or white on your scan. Okay, these typically indicate some sort of change in your tissue or pathology. If you have, because it's normally pretty uniform, then you get this bright spot and say, oh wow, there's where the issue is. Okay, and so there are sequences that have been developed. You'll hear about different sequences. And the one we're going to talk about is STIR, so you can specifically visualize different aspects of the tissue. Okay. Because otherwise it's just things occlude visualization of other parts of your body, right? So there are sequences, tricks that you can do to remove certain images so you can see kind of underneath them. That's actually what's really important in this. So this, you'll hear a lot about something we call STIR positivity, right? This is short tau inversion recovery, right? So this is one of these sequences that allows you to the visibility of the fluid. Basically, this is how you're going to see inflammation. That's what's really important in FSHD, right? And these are typically areas of concern in a tissue. So basically, you can't really see the fluid because of the fat. Okay, we're all pretty fat, and so especially if you have neuromuscular disease. So what the STIR does, the sequence, it eliminates the signals that obscure the inflammation and the edema, right? And so they're going to maximize the contrast between fluid-containing pathology and the fatty tissue, because fat and inflammation and FSHD, this is your muscle. We always talk about your fatty, inflamed muscles, but not necessarily at the same time, and that's going to be key. Okay, so STIR, right? So interesting thing, you get the MRI, and then they're going to pulse them with a 180-degree radio frequency. Okay, boom, hit them with a frequency. And so this is going to flip the magnetization of all the tissues within the imaging area. Okay, so all the tissues now flip their magnetization. And the key is they recover, different tissues recover to their original magnetization at different rates. Okay, and this is called the T1 relaxation. And the short tau, or the T1 time, is the delay that coincides precisely when the fat is at zero. You flipped its magnetization, it's coming back to where it was, and then you know exactly how long it takes when it's right at zero. Okay, and that's where you're gonna basically you're gonna pulse it with a second radio frequency and capture the image. And so now the fat molecules are essentially at zero. You don't see them. Okay, and they're nullified and they're essentially dark instead of bright. And so now you can see the water content, which is going to be your inflammation, and areas that are hyper intense in this signal are going to be where you have inflammation, and it can be right in with the fat. It's just the fat signal has been suppressed. Okay, and so basically STIR, universal marker for active pathological processes involving fluid accumulation. Okay, inflammation, injury, edema, these types of things. Okay, so that's what we're going to be talking about. All right. So this is a key paper. I told you out of the University of Washington. This is from Dennis Shaw, but Dan Miller was on this. But Seth Friedman is a key player here, and it's still working with Seth on the pig MRI, and Seth's at Seattle Children's Hospital. Okay, longitudinal features of STIR bright signal in FSHD. Okay, this actually is from 2013, came out in 14, officially in the journal. It always takes a big lag. And so the importance of this is a study that shows that moderate to severe STIR positive hyperintensitivity often foreshadows fatty replacement. So the STIR positive signal is not where you have fat, it's where you're going to have fat. And that's the key. And this is going to be what's really important for clinical trial development and design, understanding gene expression. Okay? So essentially in FSHD, right, as you get DUX4 expression, I'm going to jump ahead a few years, DUX4 expression leads to changes in the tissue, leads to some inflammation, and leads to STIR hypersensitivity, and then you can start to get fatty replacement. And actually the STIR signal is going to go away when you have full fat, but also the DUX4 signature is going to go away when you have that much fat, because DUX4 is expressed in muscle. So here's just an image up here for those of you looking on the video, and if you're not, I'll just try to describe it. This is a calf cross section, right? And looking at Intramuscular fat, which is on the left, is in the T1 cross section, is appearing dark. Right, you're looking here, this fat right here. This is just a little bit of fat kind of fibrils going through here, and then but not very much fat at this point, right? Just a little bit of fat. So this muscle here, this is essentially is the question is, what's going to happen to this muscle later? Okay, so now if you do the stir sequence and you come through and you can see that you actually get very bright stir signal right here. Okay, now these other two muscles, and this is scored a 2. Score is moderate to severe pathology. This muscle here in the center with the 0, right? You can see in the T1 cross section, there's no pathology and there's no stir signal. And the one here listed with a 1, there's a little bit of pathology. And so this is kind of a mild pathology. And you can see again, it's not very much of a stir signal. Okay. Looking down at this other figure, this is again looking at a single person, looking at cross sections down the leg, looking at the T1 and the stir signal. And what you can see are areas here. Look at this arrow here, right? You have a bright stir signal, in areas where you're starting to get some fat, into the muscle, right? But the thing that's interesting is that in areas, if you cross the top all the way over here to the right, we're in the same muscle at a different part of the muscle where you have a lot of fat, right? There's a lot of fat here, and you can see the fat, and that's this it's not as dark, but you see there's very little stir hypersensitivity, okay? So what has happened is you had the stir, so this is actually within the same, right? The stir positivity, right, happens before you get the fat replacement. Okay, that's actually, okay, that's going to be the key. Okay, so the stir positivity looks like it might be predictive of fatty replacement. All right, so that's actually what's really important to know, because if you're going to biopsy muscles and look for gene expression patterns, whether with or without therapeutic, it would probably be pretty good to know. All right. The follow-up paper on this, again, from Seth Friedman, Mark Ferguson was on this, but you'll see it's the same name, Dennis Shaw, Leo Wang, and gang, looking at this. This is MRI changes, metrics in FSHD, stir and T1. And the key here is going to be, they're just expanding on that stir positive muscles with confluent regions of fat at baseline increased more in fat, while stir minus muscles had increases in just septal fat over the study period. So they're going to look over the course of 2 years. And they're basically going to say if you have a stir positive muscle with a little bit of fat, 2 years later, it's got a lot of fat. And if you had a stir negative muscle with not really any real fat, just a little bit of this septal fat, 2 years later, it still doesn't have very much fat. Okay, so stir positivity is predictive of fatty replacement. Okay, and this is some of the data. Looking at here, it's just one individual at 0, 12, 32, and 48 months. And you can see, right, you get this stir positivity here at 32 months, a little bit of stir coming in here at 12 months, very little at baseline. And you can see that at 48 months, the stir hyperintensity is essentially gone, but now you have a whole bunch of fat. Right, so it goes increase in stir and then increase in fat over time. Okay, so this is really important for understanding, also for using MRI as a marker, as a biomarker for disease progression, but also for tracking your therapeutic efficacy. Okay, and just looking at it one more time, a stir positive muscle versus stir negative muscle. This is looking at the gastroc, and again, you see a stir positive signal at time 1. Right, very bright muscle up here at time 1, at time 2 visit, the stir signal is gone. But at the time 1 signal, when you had the high level of stir, you had just a little bit of muscle fatty replacement. But now at time 2, you got a whole bunch, right? It's very fatty, okay? So little bit of fat, high stir hyperintensivity predicts a lot of fat. And then the stir is gonna go away. Now, if you look differently at a stir negative muscle, and you're looking at a little bit of fat, but stir negativity, that actually does not become very fatty. Okay, it's just going to be, we call it septal fat. Okay, it's relatively unaffected at time too. So in the absence of stir hypersensitivity, again, which is indicative of inflammation and indicative of DUX4 activity, right? It makes sense, right? So high levels of inflammation, high levels of DUX4 gene expression, activity. I'm kind of jumping ahead of myself, gonna lead to your fatty replacement. So this is, everything we take for granted now, but this is, the work that was, it's really fantastic work that's putting this all together. All right. So now we're going to jump back a little bit about this DUX4 signature. And this paper is one that everybody should know. Okay, this is the Yao et al. 2014 Human Molecular Genetics. DUX4-induced gene expression is the major molecular signature in FSHD skeletal muscle. This, I would call this a seminal paper in the field. Okay, because back in 2014 at these FSH research symposiums that we don't go to anymore, back then, people are still arguing about that DUX4 is such a low-level gene expressor. If you do RNA-seq on muscle biopsies, it's hard to find you just don't see a whole lot of differences. Things don't really come out. There were questions about what is the actual gene still for FSHD, even though the Lemmers paper in 2010, science paper, the unifying theory for FSHD, to me genetically nailed it that DUX4 is the pathogenic gene. They were still debating about it. This paper to me put it to rest, although for us it was already put to rest. But and for some people out there, I mean, the earth is still flat. Okay, nothing you can do about it. You just nod your head, say, all right, talk about something else. All right, so what's going on here? Well, basically, this is led by Stephen Tapscott and Silver Vandermeer, their labs. What they did is they overexpressed DUX4 in human muscle cells, different types of human muscle cells. And they said, what is the DUX4 signature? Okay, and so they pushed the system. Overexpressed DUX4, and what are the genes that are overexpressed? Then they went to FSHD muscle biopsies compared to control, and they said, is the DUX4 signature, if you know what to look for, can you find it? Because that was part of the problem, right? That you have thousands of genes are misexpressed. Especially differences between people, differences between age, differences between muscle groups, differences from stress, differences from their life experiences. You take biopsies and it's just, this is a gamish. You just there's tons of genes that change anytime you take a biopsy. Okay, but the question is, if you know what to look for, can you find it? And that maybe sounds kind of silly, but it's actually really good, Instead of just looking at everything, narrow it down. Let's just look for, and so what this paper showed is that if you push the DUX4 and say, these are the genes, so there may be thousands of genes that are misregulated, and there even could be thousands of genes misregulated by overexpression of DUX4. But if you, what are the ones most significantly upregulated by DUX4 in muscle, human DUX4 in muscle, and are they present in biopsies? And what they showed is they come up with 67 genes significantly upregulated by DUX4, and in DUX4 positive muscle biopsies, DUX4 target pod. Now, part of the reason for this is you can't really detect DUX4 itself in a muscle biopsy. The mRNA for DUX4 is so lowly expressed, and the immunostaining doesn't really work in biopsies for DUX4. And so I think that's still the case. So you can't detect DUX4, so you got to find the target gene. So normally you would just look for the primary driver, DUX4, but you can't do that in muscle biopsies, right? So they said, let's look for the signature. And what they found is that in FSHD muscle biopsies, the major determinant of abnormal gene expression is the DUX4 signature, right? DUX4 target gene expression level then associates with clinical severity, okay? You force DUX4 expression in muscle, you get this gene signature. You look in FSHD muscle biopsies, and that signature is there, not present in the controls, okay? This is basically the DUX4 gene expression signature that distinguishes an FSHD biopsy from a non-FSHD biopsy. Case closed. Except for the few people who just don't think we landed on the moon. All right, Baby steps, have to keep going, right? So this actually is another spectacular paper, same groups, Stephen Tapscott, Silver Vandermeer, Robbie Twill. MRI-informed muscle biopsies correlate with MRI with pathology and DUX4 target gene expression. Well, I just told you that target gene expression correlates with severity. Now they're going to go in, and this is one of my favorite papers, right? They're going to now bring an MRI into the matter, and they're working with the same people, right? See, Seth Friedman, Dennis Shaw, they're here. Leo Wang must be. Oh yeah, Leo Wang's the first author. And Xiaoshen Wang actually is going to become prominent in this as well. And what they're going to show is that now if you do MRI and you look for stir activity, so the genes that were identified, a lot of them were involved in inflammation. We know inflammation is what's detected by your stir hypersensitivity, right? And so they're going to look at stir hypersensitivity, and they're going to find that correlates with muscles that have active disease by histopathology and DUX4 target gene expression. Okay, so now just adding to the story, we know that DUX4 triggers target gene expression as transcription factor. We know that those target genes are greatly enriched in FSHD muscle biopsies. And now you can actually find those by looking for stir in FSHD biopsies. And now you can say this is where the DUX4 genes are active, and it's where you're getting the inflammation and the pathology. Okay, so that's what's going on here. I love this figure, right? Because this actually gets directly to the key to what's going on for clinical trials and assaying clinical trials, and whether or not you use MRI informed or not informed, meaning you did an MRI or you didn't, and you chose muscles that are stir positive or not. And this is why that matters, right? So at the time, there's a 4 target gene basket: Ludex, KHDC1L, which is our serum biomarker, by the way, TRIM43, and PRAME F2. We'll talk about PRAMEs in a second. Okay, these 4 genes, and these are what I believe Fulcrum used, are called a basket of genes. It doesn't matter what they do. Bottom line is these were genes that were significantly upregulated in FSHD muscle biopsies and their DUX4 target genes, okay? And so now if you go through and down here, if you're looking online, and if now I'll try to describe it, I don't know how many here, 30 some odd muscle biopsies. The red stars are from FSHD and the blue stars are from healthy. So 1, 2, 3, 4, 5, 6, 7, 8, 9. Looks like, if I can count, nine healthy or non-FSHD muscle biopsies. And then the rest are, and what do you see? So they're going to now look at these gene expression patterns of these gene baskets, okay? So, number 1, not all the muscle biopsies express all 4 target genes. So you need to look for a gene basket. If you're just looking at 1 or 2 genes, you might miss something, okay? The other thing that's important about all this is down here at the low end, the ones expressing 0 to 1 target genes. FSHD muscle biopsies are there, overlap with healthy. You can't distinguish healthy from FSHD down here. And then you can look up here at the heat map at the top, which one is expressed and correlates up there. But FSHD and non-FSHD are mixed together. So your gene basket's important, okay? But that's key. Note some of the FSHD biopsies have low or no target gene expression. So now, if you're going to do a clinical trial and you take your baseline and it doesn't express any target genes, you're really limited. That may just be a no-go sample, right? Not going to get any information from that. Can't go down from 0. But this is the key. This is the most important figure, okay? What they're going to look at, they then went back and said, which one of these muscles had stir positivity? Which one were a stir positive muscle versus normal MRI, no stir positive, non-stir positive. I don't really know the lingo. I'm not an MRI person. But this is striking if you're looking online. If not, I'll show you. You're looking at the 4 genes. Those ones that are stir positive basically are going to be the ones that express the target genes. The ones that are stir normal, right? Very few, very low gene signature. So you're really going to push the system in your favor by taking your biopsy from a stir positive muscle, okay? So this really strongly supports using MRI stir positivity as clinical trial inclusion data. And what I mean by that is different muscle, you're going to have different levels of path, different muscles. They're going to want to, now I got to tell you, if you do RNA-seq, generally they're going to take the biopsy in a clinical trial from the same muscle, right? So there's a reason they may want MRI from you to see if you're going to have stir positivity in the muscle that they want to take, right? There's a lot of inclusion criteria, but I could, it's really reasonable to say that you need some level of stir positivity. As we showed, stir positivity correlates with active DUX4 expression. If you're looking to knock down DUX4, you're going to want muscles that are actively expressing DUX4. Stir positivity predicts that, active disease, DUX4 expression, inflammation, this subject, and it's not like it comes and goes like really fast. I mean, this is over the course of years that you're going to maintain that to get to fat. It was a 2-year window to get to see the fat replacement. Clinical trial is going to be looking at 3 months or 12 months or whatever. So you can understand, boy, you have a better chance of finding a muscle that's DUX4 positive with high level of gene expression of the basket genes if you pick a muscle that's stir positive versus one that's not stir positive. So this might be why you might not get into the trial, right? You're too healthy. Or the muscle that they want to assay doesn't have the pathology they want to check. Okay? Now this is, we worked with them on this paper. I'm not going to talk about our data, but again, Stephen Tapscott, Xiaozhen Wang, Seth Friedman, you're going to see it's the same groups, right? Really the leaders in the field. Regional and bilateral MRI and gene signatures and FSHD, implications for clinical trial design and mechanisms of disease progression. Right? So now they're going to look at Yao's 69 genes or 67 genes, whatever it was, and they're going to try to get a good, what's the best basket? So the trick was, in the Yao paper, basically they were saying, what's the difference between FSHD and not FSHD? Here's 69 genes. Right, so what they're going to do is they're going to refine the DUX4 signature and put it into baskets, either 6 genes, 12 genes, 29 genes, that may have different power, but the reality is the basket of 6 genes is pretty darn good at distinguishing FSHD and levels of FSHD, histopathology and disease activity in muscles. But different companies may use different things. Fulcrum 4 versus, so as you will see, sometimes they refer to the basket of 6 or the basket of 12. So most of these genes are expressed in testes, and they're cancer genes, they're immune genes, and embryonic genes. Because DUX4 is normally expressed embryonically when you were 4 cells old, you were expressing DUX4, and it drives an embryonic program. And if you express it in your muscle because you have FSHD, that's bad. You don't want to express an embryonic program. The other place DUX4 is expressed is in testes. And okay, so now all of a sudden you have skeletal muscle expressing testes genes because of DUX4 expression. Again, bad, don't want it. And then cancer. DUX4 is found. Now, Alexander Bellew identified DUX4 in the 99 Gabriel's paper as a gene in the region with an open reading frame. Didn't really know what it does. But the cancer folks found DUX4. It was expressed as a fusion, and it's actually expressed as DUX4 in certain cancers. Okay, so it actually predates the FSHD work, is that because it was an interesting cancer antigen. Okay, predates understanding, the confirmation that it was involved in FSHD. Okay, so it turns out then a lot of these are cancer antigens. So that's what PRAMEs are. I've never heard of PRAME. That's preferentially expressed antigen in melanoma. This is basically a group of cancer and testes antigens expressed in a variety of tumors. They also are expressed in FSHD. Okay. All right. So there's connections with cancer biology, okay, that are kind of interesting that we're not going to get into. Okay, so let's look at these genes. What matters to these? It's really, really, really important data. Okay, so this is just looking at the D4 basket of 6 genes. Okay, and looking at stir positive in red and stir negative in blue muscle. And for those of you that are just listening, what you'll see is that if you look at the DUX4 score compared to whole muscle % fat, okay, you will find that in stir positive, all of them have very high gene expression for DUX4 in the stir, and they have a range of fat from say 5% to 80%. In the STIR minus, everybody is under 20% fat. And the DUX4 signature goes from almost 0 up to, whatever. It goes up, All right. So this is actually really, really, really important. And you can look at other metrics if you're looking online, but this is the key. The key is right here, 20%. Right? Once a muscle reaches a threshold of 20% total fat, the DUX4 score is extremely sensitive and predictive, right, for pathology, okay, for active, right? So if you look at STIR positive, essentially all the STIR positive muscles have greater than 20% fat have very high DUX4 gene expression signatures, and the STIR minus, all right, actually have the whole range of DUX4 signatures, but you can distinguish this based on fat content. All the STIR minus muscles have less than 20% fat. Okay, some of the STIR positive have less than 20% fat because they're on their way to getting fat, right? But this is actually really, really important because the MRI, the STIR positive muscles all have high DUX4 scores. And once you clear the 20% fat threshold, you are you're just going to eliminate your chance of getting low DUX4 signature scores. So if you're going to screen for a clinical trial, you should be using STIR positive muscles for gene expression with an overall fat content of over 20%. And now you're not going to have low outliers that might screw you up. These are going to be your greatest window for seeing therapeutic knockdown of DUX4 gene expression. Okay, so what are we talking about? Let's just look at this now. I threw together some graphs, just like I did for Brad, but this time I thought about them a little more and made sure the math was right. And I can explain them a little bit better, and I added in some actual graphs because Takiko showed me how to use Prism. The way I make graphs is I tell my people to make me a graph. How do you make a graph? I say, Chris, can you make me a graph? Takiko. Anyway, now I can make my own. Yay. All right, so MRI-informed biopsy is designed to decrease the variability in DUX4 and DUX4 target gene expression. What do I mean by that? I just made these numbers up. This is my cartoonish interpretation of what's going on, trying to simplify it so people understand it. All right, so a regular biopsy, someone with FSHD, no MRI, taking a muscle, going to look at the basket of 6 genes. You could have a range, you could have 1, 5, 3, 10, 0.5, 6, 7, 0, 9, 2, right? The range of your basket of gene signature could be 0 to 10 arbitrarily. Huge range. Here in this case, it's going to average at 4.35, and your variability, you're going to have plus or minus 3. Huge variability. Okay. The idea is if you use an MRI-informed biopsy and pick STIR positive muscles that are on there that have active disease, right? Now it's going to be like 7, 6, 7, 9, 6, 5, 7, 10, right? Your average is going to be around 7.1, again, the way I did it, with a range of like 5 to 10, half the range, and your variability 1.14, a third of the variability. Much tighter. So baseline MRI, if you're looking online, you can see a little bar graph that I put here. Huge variability in your baseline. MRI-informed baseline, clustered more together with high levels. So this is important because if you're looking for knocking down this signature, either with a placebo or an siRNA, the test, the MRI-informed is going to give you a much better therapeutic window to look for the knockdown or to not see the placebo having any, mystery effect, right? The no MRI, if you're already down near 0, those are going to be almost useless for seeing knockdown, right? So you're losing a bunch of your samples, okay? All right, so let's do a placebo control. Placebo control, no MRI, right? Just doing a biopsy. Placebo control. Again, arbitrarily, I'm going to say, your baseline can be 1, 5, 3, 10, 0.5, 6, 7, 2, 8, averaging around 4.7. And then you do like a 3-month time point on your placebo, and you've gone 6. So this one went 6, 3, 3, 7, 5, 2, 10, 4. Remember all these. And your average is 5. Okay, so your increase with placebo, 5.88. But did that happen? Well, subject 1 went from 1 to 6. So they actually, DUX4 went up 500%. But subject 2 went from 5 to 3. They went down 40%. Subject 3, 3 to 3, they had no change, right? Subject 4 went from 10 to 7. Oh, placebo's having an effect, It doesn't, so you're looking at this, and because you're all over the map, 7 can go to 10, 2 can go to 4, 8 can go to 5, and then you're 0. You got to throw this one out. You can't use this one. Number 10, subject 10 had 0 baseline. You can't assay them. You've lost that person. They got biopsies for nothing. They're gone, right? So so on one hand, you had a 5.9% increase in the DUX4 gene signature across 10 subjects on placebo. But if you look at the individual subject chain, it actually went up 152% on average. So which one's the right one to look at? Well, I just like to see all the data. What I actually like, instead of these bar graphs, I like to see the individual graphs. An individual here, before and after a straight line to where did they go? Individual number 2 went down. Individual number 3 went up. Individual number 4 went down. Individual number 5 went down a little bit. Number 6 stayed the same. Number 7 went up. That type of thing. Okay, where did they go? And I look at this and tell you what this tells me. This is all just the noise of the random biopsy. Nothing's going on. That's what I got out of this. That's the placebo. But it's all over the map. It's all over the map. You could say, but this kind of matters. Because if you compare to this percent change when you do your drug, you might say, oh, placebo led to 150% change. My drug led to a 50% knockdown. But if I compare it to 150, maybe I'm now down at like 90% knockdown. So you can actually, these are ways that you can manipulate the numbers. But I don't care. I look at this, and if it's presented, this is why I always ask for this. I always want to see the individual. I look at this, and placebo, this is placebo, nothing's going on. All right, what if you now go to an MRI-informed placebo? Now your baseline is 9, 5, 5, 7, 5, 6, 7, 6, 8, 6. Say that. Okay, read them back to me now. Time point, 7, 6, 5, 5, whatever. So you started at 6.4 on average. You ended on 6.2 average. Your variability is 1. Overall, your change, you have a -3. You have a 3% decrease. Basically no change, though, on the individual. More important, because you're not seeing these wide fluctuations due to the randomness of the biopsy. And if you look at our untitled bar graph, then you're looking at the DUX4s on the y-axis, the DUX4 basket, gene basket. I didn't know how to change this. It's basically the same. No change in placebo. And everybody's up here where you can measure it. You have to use all 10 biopsies. And you look at the individuals, one went up, another one went down, one went up, another went down, one stayed the same. No effect. But your therapeutic window for looking for an effect is much bigger. So you have more confidence that there's no effect from the placebo. It's not doing anything, as it should. All right, so now let's put in an siRNA. So we're going to put in an siRNA, and we're not going to do a biopsy. We're just going to put in the siRNA and see what's going on. Right, so baseline can be 1, 5, 3, 10, 0.5, 6, 7, 2, 8, and 0. Well, this sucks. You got to throw out another biopsy. Your zero's got to get thrown out. That person's, at least they got the siRNA. And at your time points, 0.75, 1, 1, 0.5, 0.25, 1, 1.5, 0.51. You notice that all the numbers are pretty damn close because the siRNA is working. That's what it tells me. You went from 4.7 to 0.83, but what about the percent change? First off, everybody went down. Everybody on the siRNA, DUX4 gene basket expression level went down. That's what should happen. That's key number 1. How much did they go down? Well, number 1, only went down 25%. Is this a problem? Right? Because we saw this in a video. Someone only went down 23% or something like that, or 27%, I can't remember. Oh, it's not working very well in that person. What if it's this person who started at level 1, right? Because this is no MRI biopsy. You started at level 1 and you went to 0.75. What if that's as low as you can go? Remember, it's the number of cells expressing DUX4. So this individual might have 1 in 1,000 cells expressing DUX4, where this individual, number 2 and number 5, might have 5 in 1,000 cells expressing DUX4. Okay? Maybe you knocked it down the same, but you just didn't have as much opportunity to knock it down. So 25% and 80%, maybe they're not that much different. Maybe. Maybe not. The question is this low point here due to the fact that the starting level was so low? 0.5 down to 0.25. It's only a 50% reduction. On the other hand, you're down to 0.25. It's really low. That's a grade. You're almost off. You just didn't start that high. What about number 4? Number 4 started at 10, right? They finished at 0.5 as well. That 0.5 is actually higher than the other one, than number 5. So from 10 to 0.5 is a 95% knockdown. But the actual DUX4 levels are higher than in number 5 that only saw a 50% knockdown. But it doesn't matter because it's working, and it's working really well, right? But you have this tremendous variability potentially because of the randomness of the biopsy, right? Okay, let's look at the bar graph, and you see this great variability here in the baseline siRNA and then the siRNA 3-month. Everything crashes to 0. That tells you it's working. That's what you want to see. Look at our individual time points. It doesn't matter where you started. Everybody's going down and everybody's going to 0. That's working. It's just that this percent change on a particular individual can be kind of off. All right. What happens then if you remove some of the low outliers, right? So here we had an overall change, right, of 82.35%. We're going to remove the low outliers, and actually it's going to go up, right, to 84, a greater change if you remove the low outliers, right? But again, if you have a robust response, it doesn't matter. And I'm not that fussed about a couple low changes, is because their baseline is very low. Okay, so let's then go down here, and we're going to look now at no MRI biopsy, remove the low outliers. What are we talking about? Right, we're going to remove number 1, number 5, and number 10. Now you're only looking at 7. You see that everything kind of tightens up, and you get more therapeutic window, right? You get more therapeutic window. But again, doesn't matter. It's working. Now if we go to no MRI biopsy with some high outliers, okay? What if we actually instead, you had some people that were very, very selected arbitrarily better, the 10 that you selected, with baselines of 10 and 9, right? You're still getting down to 0.75 and 0.5 and 0.25. You're getting 90% greater knockdown, right? It's working really well, right? But these outliers, they're actually driving the percent change, making it 90% change because you have a few very high initial ones. But that's probably pretty good, right? I'm not saying it's fake or anything. I'm saying they're driven by, this change is driven by these high baseline subjects, okay, versus the others. Everybody's change is about the same. Everybody's getting pretty darn close to 0, okay? All right, so what if it's not that good a drug? What if it's not that robust a drug? Well, what's it going to look like? Well, again, if you have no MRI, your baseline is all over the map, but your changes are going to be not so much, right? Your second time point, maybe you go from 1 to 0.75, you go from 5 to 4, you're still going down, you go from 3 to 1, you go from 10 to 3, that's a pretty good one, that's 70% knockdown. 0.5 to 0.25, that's a 50% knockdown, right? You have 7 to 4. What if you go from 2 to 4? Did that work? Why would one go up? That doubled. But actually,
the other way to look at it is everybody that's on siRNA is at 4 or lower at the end. And maybe you just happen to pick a spot that was really low at the beginning. And actually going up here, which on one hand you might say everybody should go down. But if your drug doesn't work that well, maybe somebody went up, but they didn't really go up. They just started too low, right? Because of where the biopsy was taken arbitrarily. And actually the drug is actually still working. Just not a very great drug, just not very good. Right? This grad student didn't make a very good siRNA. We finally found one, right? All over the map here, right? Your individuals, again, I always want to see the individuals before and after. What's going on? That tells you. And what this tells me, especially looking at the levels, is there's like a, the therapeutic threshold here. Everybody gets below 4. And, I'd say, this is probably still working, just not very well, even though somebody went up, surprisingly. Now let's go to MRI-informed biopsies. MRI-informed, right now your baseline, everybody's above 5. Your average is at 6 and only a variance of like 1, right? After the drug, you're at 0.9 and the variability is 0.4, plus or minus 0.4. I know I'm using the wrong term sometimes, sorry about that. Well, let's look at our bar graph. Very clearly this is working. You have much less variability at the beginning and everybody's crashing down to 0, right? If you look at your individuals, everybody's going down to 0. This is a very robust, efficacious treatment. This is working really well, okay? You have your 10, a lot of them are on top of each other, and everybody's going down, okay? That's what the MRI-informed biopsy is supposed to give you, this better window, right? So it's not overlapping like here, right? So let's see if it's not as efficacious. Let's see if it's not as good like the previous one. We'll take the previous drug. But the thing is, everybody's baseline is high. And so even if your drug isn't as good, even if your siRNA is not as good, everybody's going down. Everybody's, you have one here at 5. 5 is even higher. It's not that great. You have some at 3.5, but you're still going down because you started at 7, you didn't start at 2. Right? So now when you look at the individual thing, everybody's still going down. You have a better chance at figuring out if your drug, which, is working, but not working as great as you maybe hoped, but it's still working, but you'll find it out by using the right gene basket and the MRI-informed biopsy. That's why you want to do it. Let's look at a small molecule. Hypothetical small molecule. We're going to go through and go through, not going to use MRI-informed biopsy, not going to use stir positivity. What could happen? What could go wrong? Here's 20 subjects in this case, if you're not looking online. And the range is from essentially 1 to 10. Your baseline. Your average is 5.75 plus or minus 1.75. And after 3 months on the drug, you're at 4.5 plus or minus 1.8. So on average, your change, you're going down 21%. On average, drug's working, right? Well, it's trending positive. It's looking promising. It's showing possible biological activity, or repression of biological activity. It's a breakthrough. Donate more money. But is it? Well, let's actually take a look. Okay, it's 20% change. It's trending. These are terms. Watch out for these terms. Trending positive. Looking promising. Suggestive of biological activity. If we look at our baseline compared to our 3 months, if we do this with here, what do we got? Well, 20% change. There's an awful lot of overlap. I call these the swarm of bees type of graph. A swarm of bees here and a swarm of bees here. And the average of the swarm changes. But that's not what actually matters, right? What actually matters is not the average of the swarm, right? Because I look at this, I say, no, wait a minute. You know me, I like to look at other things. So what are we going to say? What you talking about, Willis? Damn right. What you talking about, Willis? We say that in lab all the time. When we get a good result, we say, that's what I'm talking about, Willis. But this ain't a good result, right? Arnold's going to look at this and say, show me the individual data. So Arnold Jackson's looking at this saying, wait a minute. Yeah, on average it's down 20%. But this guy goes way up. This guy goes down. Actually, it's all, if you look at the, for individual percent change, it's actually an increase of 12%. Right? The down is driven by a couple that go down far that are mitigated, you know, on the percent change here. But on the individual change, it's actually the drug's having no effect whatsoever. But we got an IPO coming up, so we're going on to phase 3, hypothetically. One could imagine. Can you imagine someone doing that? Interesting world. All right. Mouse data you got to look at differently. Mouse data, let's look at the human xenograft data. Here's human xenograft. We're going to grow FSHD cells preclinical data and look at it. Grow a mouse muscle. We're going to grow the muscle in the mouse, the human FSHD muscle. It's going to innervate, going to vascularize, it can contract, and we're going to treat the mouse, and we're going to look at its muscle. Now, the problem is we have to assess the entire muscle. So what that means is that you don't have a before and after like you do in human trials, right? What you have is you have muscles for mice that are untreated and muscles that were made the same way at the same time in mice that are treated. And so now you can't compare, right? So if we're looking at the xenograft model, now the problem with the xenograft model is it's like your uninformed biopsy. The range is all over the place from essentially 0 to 10. But your treatment should all crash down, right? Your treatment should all crash down to 0. But you can't do the individuals because you can't say that this individual here became this one here. The high point in the vehicle is the high point in the treated because you have to use the whole muscle for the analysis. You can't do the experiment the way I like to see it. So you're just stuck. And that's how you end up with data, xenograft data looks like this. So we don't know, is this actually a 90% knockdown from a really high starter or not? That's why you just have to do the groupings. You're stuck with it. What about the FSHD flex mouse? Here's what the FSHD flex mouse looks like. Tamoxifen administration has a tough time, just like some of y'all might have a tough time. He's expressing DUX4 in his muscle. Those are his siblings that don't have FSHD, just having a good old time running around. And but he's, you know, the FSHD-like mouse is, you know, having trouble getting around. Maybe you can relate, right? But now the problem is we have the same issue. We have to assess the whole muscle. But this is a little bit better. Because it's a transgenic inducible model, we can force the system and make it more like an MRI-informed biopsy because we can actually push the system with tamoxifen to induce DUX4 expression, and now everybody's starting high. And you still have vehicle versus treated, and you can't do the individuals and compare them, but you can have more therapeutic window, and you can see much better that the drug is working. The difference is the xenograft model is the true human genetic situation in FSHD, how it is in your cells. It's actually human cells in the mouse. So that's what the value of that is giving you. What you're getting here is you're showing more of the therapeutic window, okay, that's working when you force expression of DUX4, and you can tell how good it's working, okay? It's a little harder to tell how well it's working in the xenograft because it's like an uninformed biopsy. All right, we're going to stop the share. All right, stop the share. So there we go. Crystal clear, right? Again, it gets back to the devil's in the details, right? When you're looking at the gene expression data, are they doing MRI-informed, STIR positive muscles that they're biopsying, right? Because if you're over 20% fat and you're STIR positive, you're going to have high level, you have active pathology, active DUX4 expression, you're going to have your inflammation, and these are going to be at the high end, and you're going to be able to see a great therapeutic window for knockdown. Or are they taking random biopsies, where DUX4 could be 0 because you have so much fat already that the DUX4 expression is not there anymore, in which case you won't see knockdown. Or it could be DUX4 could be at 1 or 2 because it's pretty healthy muscle that hasn't even started the progression down to pathology to fat. In which case, again, you're not going to see any therapeutic window, almost like throwouts, right? Now, if you're in the trial and you get the siRNA, you still potentially get the benefit. But for informing the metrics on whether or not the drug worked, it's not that informative. Okay. The other thing is, how do you compare? I showed you in the mouse, you can compare vehicle to treated. Sometimes that is actually what's done, even in clinical, right? Are you comparing individuals before and after treatment? But you can also compare treatment to placebo. And if you do that, because placebo, as we showed you, depending on how you do the numbers, if placebo looks like it's going up 100% because of where things started on baseline, not really going up 100%, but you can make the math look like it's going up 100%, and you're going down, you can actually show that versus placebo, you have a much greater effect than versus before you start. Does it matter? It probably matters to investors. FDA is going to force you to show them the real numbers. It matters in press releases, I suppose. What I get out of it is that it works. All these siRNAs work. What's it going to matter? Does it matter whether it's 80%, 85%, 87%, 70%? What I want to know, durability of the effect, durability of the knockdown, targeting because of potential adverse effects and toxicity, cost in manufacturing when you're making it, delivery IV versus sub-Q. Put it all together. A lot of those things are more important to me than whether or not you have 80 or 85% knockdown. Would I like 100%? Sure, give me 100%. Throw that in as a bonus. But to me, the most important thing, again, now I don't want a 20%. Now, if you tell me 20% knockdown, it's going to have to be in everybody 20%. I don't want that so much. Okay, that's different. But if it's 20% in an individual, that doesn't mean it doesn't work. Because you got to know where that individual started. Right? Anyway, you can see the devil's in the details. I like to see before and after the specific individuals. This holds true not just for siRNA, for gene therapy. When CRISPR presents their data, I would like to see the methylation before, methylation after, DUX4 before, DUX4 after, all these things. That's how we do it. That's what we're going to show, except methylation, because it ain't going to change. But DUX4 before, right? You take a biopsy beforehand, biopsy after, right? Now, some of this might get mitigated a little bit by circulating biomarkers, right? Circulating biomarker, now that's going to be systemic, whether the DUX4 is going down. I'm not 100% sold on the circulating biomarker, I have to say. Still, the verdict's out. I've talked to some people that have suggested that and they don't see it as robust as Avidity sees it. But we'll see. I hope there's a circulating biomarker. Last thing before we go, I do want to talk about biopsy for a second. I always say, right now we have MRI. We're moving hopefully towards circulating biomarkers and whole body MRI. But when you're targeting DUX4, and you're going to want to show target engagement and reduction of the pathogenic driver, the DUX4 signature. We can't really show target engagement, but you infer target engagement by the reduction of the DUX4 gene signature, right? People need biopsies. Devil's in the details again. Ask. Don't let requirement for a biopsy put you off completely. Ask. How many cores are they taking? These are needle biopsies as opposed to open biopsies. The biopsies it's not as much as you think when it's a needle biopsy. In fact, sometimes that's the problem. It can be very little, especially if it's not MRI-informed. It can be all fat. Fulcrum did 4 cores. They put the needle in one spot, but they went 4 times. That really upset some people because essentially you're getting 8 biopsies, even though they tell you 2. Find out. Ask. What do you mean by biopsy? That's your right. Ask. You might even add, they should be able to show you how much. It's actually surprising. It's like a little spaghetti string. It's like a red vine thing. It's not as much muscle as you think. You're not going to have your whole muscle collapse afterwards. They're not going to take it if you have no muscle. They're not going to take it if you have too much pathology, right? Hopefully it's going to be stir positive, but relatively healthy on its way. And you're going to need to take it. And that's honestly what needs to be done now. I don't like it. You don't like it, but it needs to be done. Don't let that put you off. If you otherwise, you can be in a clinical. Now it's all up to you. It's not as bad as it sounds. Many of the MDs that have done it on themselves. But if you don't want to do it, that's fine. And you want to wait. I understand. I volunteered for one. I was happy I didn't have to do it. They said, oh, my genetics suck. I wanted to experience so I could tell people. It's easy to tell people about biopsy. But I don't want, just the word biopsy makes it sound, a lot of people even had diagnosed by biopsy back in the day. Okay. So anyway, it's not an open biopsy. It's a needle biopsy, and it's really important. And the important thing is that now the technology is there to make sure they get the most information out of it that's beneficial to you and beneficial to the field. But if you don't want to do it, someone else will. So you can hold out, and that's fine. Nothing wrong with that. But ask questions and find out. Make sure you understand, so you actually properly are informed on how much material, what the biopsy means. Just be informed. And those of you that are in these trials and doing all of this, well, fantastic. Thank you so much. Everybody in the field, thank you so much. You're really, really taking it for the team, and you deserve to be on a good drug for life. I hope that's what people get. Check out your Helsinki things. Rights. Inform yourself. Ask questions. Okay? Trials are, we got some good stuff coming too. If you didn't get into one of these trials, there's better stuff coming. I shouldn't say that. Potentially better stuff coming. I like the Arrowhead Sarepta one pretty good right now. But there's different stuff coming. We'll see. And again, I want to see manufacturing and costs come down, and I want to see sub-Q. And you know what? If Avidity is 6 weeks and Arrowhead Sarepta is 10 weeks, I'd like to see that. Let's go for 16 weeks. All right? Let's go for twice a year. All right? Set the bar high. Or gene therapy. Maybe just once. Of course, that has different risks. Okay. Got a special talking heads for you.
You may find yourself living in a shotgun shack, and you may find yourself in another part of the world, and you may find yourself behind the wheel of a large automobile. You may find yourself in a beautiful house with a beautiful wife, and you may ask yourself, well, how did I get here? Letting the days go by, letting the water hold me down, letting the days go by, water flowing underground, into the blue again, after the money's gone, once in a lifetime, water flowing underground. And you may ask yourself, how do I work this? And you may ask yourself, where is that large automobile? And you may tell yourself, this is not my beautiful house. And you may tell yourself, this is not my beautiful wife. Letting the days go by, let the water hold me down. Letting the days go by, water flowing underground, into the blue again, after the money's gone, once in a lifetime, water flowing underground. Same as it ever was, 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 same as it ever was.
Same as it ever was. Same as it ever was. Yeah. Terrible.
Okay, we're back, and as you can see, I've changed locations. It's Saturday, so I'm in lab doing saliva DNA genomic preps, as I do. And so that was the Talking Heads. Well, it was Kermit singing Talking Heads. I'll tell you where that came from. I just got back from Boston, and Went out for dinner with one of my few friends, Dr. King from the University of Massachusetts Medical School, and he does all the computational analysis, programming, and statistics, everything for our research testing. And he's actually a corresponding author on the paper that'll be coming out pretty soon. And he recommended for dinner this place called Temple Records, which is a bar right near Boston Common, which is a vinyl bar, not the kind of vinyl our CRISPRi goddess wears, but the kind of vinyl like records, actual records, and they play. And it came up because he said, well, he went there after a David Byrne concert in Boston. I thought, oh boy, I haven't done Talking Heads. So that's where that came from. All right, speaking of Boston, so now what I'm going to do is I'm just going to go through and give you my presentation. I can't link in the recording of my actual presentation at American Society of Gene and Cell Therapy, but instead I have the presentation and I'm just going to present it to you as I did. You can see what I presented if you're interested. All right, we share the middle and all right. I guess I'll just do it as I did it at the meeting. All right. Well, thank you for inviting me to give a talk. It's good to be back in Boston. We'll be talking about work that we started actually when we were at UMass Medical School in Worcester. That is on our CRISPR inhibition gene therapy for FSHD. For those of you who don't know us, we're a husband-wife team functioning as co-PIs. And I always like to say, and it's absolutely true, Takako is the brains and I'm the mouth. So I have her here on my cell phone. She's watching back in Reno. It's 5:30 a.m., but she's watching so that I can ask her questions by text. All right, first let me show our financial disclosures. Takako, myself, and Carice are founders of Renogenics, which is a company aimed at taking our CRISPR therapeutics to clinic. So just so you're aware. All right, so FSHD is a dominant gain of function disease. Essentially, what's going on is in FSHD, you're making the DUX4 mRNA, whereas in control cells, you actually don't express DUX4. So both FSHD1 and FSHD2 express DUX4 due to an epigenetic dysregulation of the normally silenced locus. And so therapeutically, you need to get rid of the DUX4 mRNA. And this is ideal for a lot of technologies that are currently in play in the therapeutic space, such as siRNAs, antisense oligos, PMOs, short hairpin RNAs, and microRNAs. Right? These are going to target the DUX4 mRNA and hopefully eliminate it and thus stop the initiation of further downstream pathology. So this is essentially an ideal disease for this sort of antisense technology because you really don't need DUX4 expressed after early development, right? DUX4 is normally a developmentally expressed gene when you're about 4 cells old. And so as far as we know, adult somatic cells don't need DUX4. So you can get rid of as much as you can, and the more likely the better. Now, alternatively, an approach, since I'm a gene regulation person, would be to shut down expression of the DUX4 gene, stop the expression of the mRNA. And this can be done either through CRISPR inhibition, CRISPR editing, this actually base editing, but it's very particular on how you do it. You can only target specific sequences, or small molecule approaches to shut down the gene. And actually we have all three of these going, but these are going to be slightly different because instead of, and the other approach is where you're still making the mRNA and you constantly have to be on the drug. Here we're hoping to just shut down the DUX4 gene. All right, so FSHD is absolutely ideal for CRISPR inhibition because there aren't that many diseases where the therapeutic is to shut off pathogenic gene expression. In fact, most of the muscular dystrophies, as you all know, are caused by defective structural or signaling genes that need to be replaced. So FSHD is absolutely, this dominant gain of function disease is ideal. And one thing that's very important to keep in mind is the target tissue is skeletal muscle. And skeletal muscle is largely quiescent, meaning it's not really replicating, okay? And so that actually is going to have implications on how you can shut this off and how stably you can shut this off. All right, so as I mentioned, FSHD is caused by epigenetic dysregulation. So each one of these triangles is a D4Z4 repeat unit that's 3,300 bases of DNA long. And, there are multiple D4Z4 arrays, head to tail, throughout the genome. The ones of interest in FSHD are located on chromosome 4 and chromosome 10. But with all of these D4Z4s around, there's only one that actually causes FSHD, the distalmost D4Z4 repeat unit on a contracted array in FSHD, and then can only when cis with a specific distal element. Okay, so this contraction leads to an epigenetic dysregulation where this distalmost repeat is epigenetically on or capable of expressing DUX4, and all the other D4Z4 arrays on chromosome 4 and 10 and throughout the genome are epigenetically silenced. Okay, so there's only this one area that you need to actually target. So if you're thinking about genome targeting using CRISPR, what CRISPR essentially is a very specific way to target a sequence in the genome. So the sequence you'd want to be targeting is within this last D4Z4 repeat unit. Now in CRISPR, one of the big concerns is what we would call off-target effects, right? What happens when your CRISPR function targets genes that are not what you're interested in? In FSHD, the problem is a little bit different. One of the concerns is the on targets, right? You have all of these D4Z4 repeat units are identical, and the chromosome 10 ones are 98% similar. So you actually cannot eliminate the on-target specificity of the CRISPR binding to each of the D4Z4 repeats. And this is precisely why CRISPR base cutting, CRISPR DNA editing, does not work, because you can't get rid of the on targets. This will just cut this all up and lead to cell death, right? The other thing that was an initial concern was most CRISPR technology, in fact, all prior to our work, was done on single copy genes where you have single guide in a gene that you're targeting. And it wasn't clear that you could actually use CRISPR to target the repeat genome. Can you actually target a sequence that's present thousands of times in a cell? And nobody knew. But hey, we went ahead and gave it a try. So the question is, can CRISPR inhibition repress the pathogenic DUX4 D4Z4 repeat unit? So conceptually, what we want to do is drop an off switch using CRISPR targeting. Instead of cutting, we're going to bring a transcriptional repressor in here and epigenetically or transcriptionally silence this dysregulated repeat unit. Now, all the other sites are also going to get the CRISPR, but that's okay because you have repression of DUX4 here. All the others are already off, so if you drop more off switch on them, they just stay off. So you actually haven't done anything bad, okay? And this is why it's ideal for CRISPR inhibition. All right, so we actually showed this proof of concept back in 2015 and 16. We're actually the very first neuromuscular disease to show that CRISPR is applicable to it, and it was very nice. We got some interesting press. And since that time, though, the question has been, can we actually take this to the clinic? Okay, and this actually came from Chip Wilson and Neil Camarda. This is actually how we met them, right? Chip had seen a TED talk on CRISPR, and he thought, wow, this is really powerful technology. Is there anyone doing CRISPR in FSHD? And so Neil was looking around, and of course, we're the only people who had published on CRISPR and FSHD. And so Neil had seen Carice give a talk on our CRISPR inhibition at a meeting. And so they called us up and came to Reno to visit. And they said, all right, so this is interesting. Can you actually get to clinic? Are you just a bunch of academics, or can you really, do you have any plan to take this to clinic? And honestly, at the time, we didn't really have any plan to take this to clinic. We're a group of 3 people. We don't really have much money. And we just figured, well, someone else will take it to clinic, but they said, well, could you do it? So we thought about it and we thought, okay, well, what do we need to do? Well, the first thing we need to do is get everything into a single AAV, right? Our academic CRISPR inhibition system was 2 vectors, 2 viruses, right? We would use the Cas9 virus because it's so big with the regulatory cassette, and then the guide in a second virus. And that's not going to clinic. You have to get everything into a single one. So how can you do this? So Carice, who's our CRISPR goddess, and this is why, she looked at the sequence and said, well, the CK8 regulatory cassette, this is what drives expression in striated muscle. That's actually pretty big, so we can make that smaller. And she said, well, the Cas, SP-Cas, now there's a lot of different types of Cas. That's pretty big. We can use a smaller Cas protein,
right? And then the repressor crab domain. Even though the crab is pretty small, she surmised that she can make an even smaller one. And if she made all of this small enough, she can fit the guide RNA cassette into the same virus as the Cas. Okay, so starting, it's actually really cool stuff. So the first thing that she did, she came from Steve Hauschka's lab and did this all on her own, and knows these promoters very well. These are the promoters and regulatory cassettes that are currently used in muscular dystrophy gene therapy. Most people use CK8, TMCK. These are actually quite large. Now remember, in AAV, you only have about 4,200 bases of DNA between the ITRs that you can actually use. So you're actually using 20%, close to 20%, maybe like 18% of your capacity just for the regulatory elements. Now, if you go to CK8E, which is the smaller one that a lot of people now use to save space, now you're only using about 10% of your capacity. Okay, this is the one that a lot of groups use. But the thing to note is, instead of having 3 copies of the enhancers that are going to drive expression, CK80 only has 1. That's actually the difference. Right? They didn't really make it smaller. They just reduced the number of copies of the enhancers. So you have less power. It's less powerful. Okay, so Carice looked at these and she said, I'll tell you what, we're only interested in FSHD. And FSHD, the heart is not affected. And so, we're going to make an FSHD specific regulatory cassette. So she's going to take out the CARG site and the AP2 site, which are required for expression in cardiac. And then she's going to put together these other elements in a very intelligently designed way. Okay. So this is real intelligent design. And she put them together into 2 sequences that we're going to call no heart and have a little heart. And the only difference is whether or not you have 2 right E boxes or you include the left E box. And importantly, these are 3 copies of the enhancers. And even with 3 copies, they're really quite small, 378 base pairs. Okay, now do they work? All right, the answer is yes, they work spectacularly. Okay, so here we are. Then we're going to show you that data here. We're going to take our FSHD optimized regulatory cassettes, the no heart and the have a little heart. We're going to fuse them to mCherry as a reporter and just see where are they expressed systemically when systemically introduced into a mouse. Okay, so minimal cassettes provide a range of expression. So these are live mice that we're imaging, and you're able to do live imaging and visualize the mCherry as it goes through. And what you can see is that they express very well in skeletal muscle, but at different levels. And the have a little heart is a much higher level than the no heart, and the expression persists over time. If we actually then dissect out the muscles and look at the tibialis anterior, the gastroc, or the quads, the soleus, the EDL, the diaphragm, what you see is we get actually great expression. Have a little heart is extremely high expressed. No heart is not expressed nearly as high as have a little heart. It doesn't look like you get much in the diaphragm, but actually that's because the diaphragm is incredibly thin. Turns out you actually are getting great expression in the diaphragm. Same thing for the EDL. You're getting good expression. It's just such a small muscle. Importantly, though, you're really not getting expression in the heart, and only a little bit in the have a little heart. And this is important because high expression of transgenes in gene therapy in the heart leads to cardiomyopathy and other problems. So this actually is a much better situation. Let's just look at it a little bit better. Have a little heart, as you can see, soleus and EDL do express very well. Importantly, the liver, non-muscle tissues don't express basically at all. And I could show you the kidney and the testes and all these other tissues. The reality is it's very, very specific to skeletal muscle and a little bit in the heart, just a little bit in the heart. Okay, so we figure this is ideal for FSHD. Okay, also, importantly, it's ideal for about 17 other neuromuscular diseases, right? That need a little bit of expression in the heart, but high expression in the skeletal muscles, but not too much expression in the heart. And the compactness of it allows you to have much more space for your gene replacement therapeutic. All right, so now we're going to look at viral genome quantification, because you might say, well, maybe it's only expressed a little bit in the heart because not much went there. We used AAVrh74, which is not a great virus, because a lot of virus goes to the heart,
a lot of virus goes to the liver and a lot of virus goes to the kidney. So even though we have 86 times the level of virus going to the liver, we don't detect any expression. 7 times going to the heart, we detect very little expression, right? Testing, but in the skeletal muscles, even though there's most of the virus does not go there, we get very high transgene expression. Think about this. If you're using RH74 in your gene therapy trial, very little of your RH74, everyone in the room knows this, is going to your skeletal muscle. Most of it is just going to your liver and your kidneys. All right, so now let's look at the repressor, right? The crab domain is 186 bases. That's actually already pretty small, right? But it has some issues. Crab domain is not wonderfully specific, and I'll show you that in a second. So what Carice did is she made 4 other minimized repressors. She used the methyl CpG binding protein 2 transcription repression domain, heterochromatin 1, HP1 alpha, HP1 gamma, and the histone methyltransferase domain from SUVAR39, and tested those. And turns out they all work. I'm not going to show you the data, but we decided that we liked the MECP2 TRD the best. It's the smallest, but it's also the most specific. Okay, and that'll show you that right here. What we're going to do is do RNA-seq on cells expressing the CRISPRi TRD or the CRISPRi crab. And now the blue and the red show you the differentially expressed genes, but those are all basically virus induced proteins. Those are due to the viral infection, not due to the CRISPR protein, not due to the Cas9 TRD fusion. Okay, the yellow dots are actually the ones that are unique to the repressor. And you can see the crab domain. Now the thing about it is you gotta remember, these repressors are targeted through the Cas9 and the guide RNA to this gene of interest, but they are also proteins that function on their own independent of the Cas9 that they're fused to. And what this shows you is that the crab domain actually can silence genes and independent of the Cas9 and the targeting. And so you get all of these off target. And so we look at this as not being very specific, and so this could be causing potential problems. The transcription repression domain in HP1 alpha, no off targets, very specific. And for other reasons, we chose to go forward with the TRD. Okay. The third thing you can do is change the Cas. So, we're just able to find there's SA Cas9 is much smaller. And so if you put in HLH, SA Cas9, MECP2 TRD, and then the whole guide RNA drug telling this where to go to the D4Z4 repeat on the DUX4 gene, it's only 4.2. You have 4.4. It only has 4.2 kb, fits very well, and it works. How do you know it works? Well, let me show you. So if we go into FSHD primary muscle cell culture, and we look at our 2-vector system or our all-in-one system, and we look at DUX4, actually look at the DUX4 mRNA, and what you can see is that we're getting, about 75% repression of DUX4, and there's different levels of repression. You can see the all-in-one, it comes in very tight. The DUX4 target genes, actually, they all go down as well. So the all-in-one vector works at least as well as the 2-vector system, if not better than the 2-vector system. If we then look at the off targets and looking at healthy cells that are either immortalized or primary myotubes, you can see that there's virtually no change to the healthy muscle transcriptome. So this is very specific, okay? Then if we go into FSHD1 cells and we look at the transcriptome, now DUX4 induces a lot of genes and DUX4 represses some genes. Genes that are induced by DUX4 become repressed, that become red, and genes that are repressed by DUX4 become induced, okay? Essentially what we are doing is we are returning FSHD1 muscle cells to a healthy non-FSHD transcriptome. Same thing for FSHD2, returning to a healthy transcriptome. Okay, so now let's go into a mouse model, right? The TigerGo made the FlexDUX4 mouse, which has the human FSHD, the human DUX4 gene inverted and driven by the ROSA26 promoter. Now, the key to this is there is still a guide sequence for our CRISPR inhibition present here, but it's only going to be in one copy, okay? In FSHD, of course, it's going to be in multiple copies, plus you're going to have these copies on these other D4Z4 repeats that are in the other chromosomes. And the DUX4 promoter is not going to be as strong a promoter as the ROSA26 promoter. So we're actually going to test this against a much stronger on switch and only a single copy and see what happens. Okay, so here's the mouse. You're not familiar with the mouse. It's tamoxifen inducible. When we add tamoxifen, the DUX4 gene inverts genetically, and you start expressing DUX4 in a set of cells. And it's dependent on the amount of tamoxifen. You can see this guy's having a lot of trouble walking around. Right, this is just a few days after induction of pathology. And these are his siblings just running around doing just fine. Okay, so now we just need a virus to put it in there. Okay, so what we're going to use is we're going to use a next generation myotropic AAV. AAV9, AAV-RH74 are just outdated. You need to use too high a viral load. They're dangerous, and there are much better things available. We actually screened the AAV myos, the myoAVs, and some next-gen vectors that are not publicly available and settled on a really good one. The key is they transduce skeletal muscle really efficiently, low immunogenicity, low tropism behind skeletal muscle, meaning they're not going to go to the liver, so they're liver detargeted. This reduces viral load, reduces cost, reduces liver toxicity, and increases safety. Okay, so you have to be crazy to go forward with AAV-RH74. All right, so what are we going to do? Well, we're going to do a systemic injection of our CRISPR inhibition in its next-gen virus at 1e to the 13 or 5e to the 13. Now remember, the Duchenne Elvidis gene therapy that is RH74 is 1.3 times e to the 14th viral genomes per kg. Okay, so we're well below that. We're tenfold below that, or less than half of that. So a much safer dose with our next gen. Going to put those in. We're going to add tamoxifen, then run them on a treadmill and see how they're doing. And what do we find? Well, without adding any AAV, the mice after tamoxifen, a lot of muscle dies and they lose weight. At the 1e to the 13th and the 5e to the 13th, they maintain weight pretty well. But as for treadmill, actually, the 1e to the 13th more than doubles their treadmill time. And then the 5e to the 13th, it goes in quadruples, right? Instead of under 4 minutes, they're up at 16 minutes. So they're able to run much better. DUX4 target genes in the mice go down in a dose-dependent manner. So dose-dependent increase in functionality and decrease in DUX4 expression. So again, the key is, we're doing this in a case that is testing even greater the ability to silence, because we're using a stronger promoter and a single binding site, which is not the case in FSHD, where you have a weaker promoter and multiple binding sites. So we're actually really pushing the system, and we have an inclined treadmill. We are pushing the system hard to see if this works, and it's still working really well, because we don't want to go to clinic with something that doesn't work very well. But that's an artificial system. Let's go into a mouse, which is still artificial, but a little bit less, because we're going to use the human FSHD xenograft model, where we take human FSHD muscle cells and put them into a immune-compromised mouse, and we're going to grow a human FSHD muscle in the mouse. And we do this different than a lot of other groups. We actually grow the muscle first. We electrostimulate it, get it so it can contract. It's innervated, vascularized, and the muscles are healthy. It's an FSHD muscle in the mouse that's functional. Then we do the systemic administration of our CRISPR AAV CRISPRi, and then we do the sampling. And so this is the same genetic situation as in you, and the question is, does it work? And what we find is just what is typical of FSHD muscle. Great variability in the level of DUX4 and the apps just on its own, just DUX4 in the FSHD muscle. But when we add our CRISPR inhibition, it all crashes down towards zero, okay? And so same thing with the target genes. So we are getting great repression even in the FSHD situation. Now that's a mouse, and we've done cells, and we've done mice, but a lot of things fail and go into trial. And what we really want to do is CRISPR inhibition in you, right, in a person, right? So in order to do this, we decided way back in 2019 when we were, after meeting with Chip and Neil and saying we're going to go to clinic, we said, well, you know what, myotubular myopathy and Duchenne muscular dystrophy, the two great programs for gene therapy at the time, they all had dog models. So we need a large animal model for FSHD. But we're never going to find a naturally occurring FSHD dog because they don't contain DUX4, right? So we decided we had to make one. So what did we choose? We decided to make a pig, okay? So we did some work with Stephen Tapscott's group, and we looked at the pig transcriptome in response to DUX4, okay? We made some pig cells, Then Stephen put in human DUX4 into the pig and compared it to the porcine version of DUX4, which is actually DUX-C, okay? And so the DUX-C, which functions in the pig as DUX4 functions in humans, its binding site is very similar as DUX4's binding site in pig. Now, why do I bring this up? So mouse has an ortholog that functions like human DUX4, basically expressed as an embryonic driver of development, but very different. So when you put human DUX4 into a mouse, you get very different downstream genes, okay? But here in the pig, you're going to get very similar downstream genes because the binding site's the same. That's actually what Stephen's group found out, was that the human DUX4 versus the pig DUX-C is very similar. And so you're going to be able to, you're activating the protein coding genes, the repetitive elements. And so essentially you're going to be able to assess the same gene expression profiles as you do in humans in clinical trials. So it's much more human-like. It's also going to be much more relevant for downstream applications. You know, once you look downstream of DUX4 in the mouse, you're playing with fire a little bit, because what you're really getting is a muscle that is expressing a foreign protein that activates many different genes, which may give you a myopathic phenotype. In pig, you're activating the same genes that you activate in human, so you're going to get a much more FSHD relevant myopathic phenotype. And that's going to be very important for assessing whether or not the response to your gene therapy is going to be okay. Okay, we used the exact same genetics that we used for the mouse, though. We had to put in the human DUX4 gene in an inducible manner. We did this with exemplar genetics. Toby Franks at Exemplar engineered these cells, and we were successfully able to make tamoxifen-inducible FSHD-like pigs, and this is what they look like. These are actually Gottingen mini pigs. They're only, they're very small. They max out at about the size of a golden retriever, but they're pigs. All right, so what does it look like? Well, when we induce DUX4, we get very amazing FSHD-like pathology. We get tons of immune cell infiltration, we get tons of fibrosis, and we get fat infiltration into the muscle. Okay, this is actually what FSHD really looks like. Mice are not very fibrotic and mice are not very fatty. Okay, so this is a much more FSHD relevant phenotype in the muscles. What about running on a treadmill? Well, these pigs, this is a pig with a little bit of FSHD. Oh, you can see he's falling. He's having trouble running. Maybe this is how you guys are on the treadmill. A pig typically would have his nose right up here and not able to fall and can go all day. But you're going to see he is stumbling and his gait is actually affected. What about over here? We can actually do force plate analysis. So we can actually measure the force and see the decrease in force on dorsiflexor torque on the pig. So we're able to assess the phenotype in the pigs. What about MRI? We talk a lot about MRI. Well, it's going to be a great biomarker in FSHD clinical trials. And if you have a full body MRI, you might find that many of your muscles have turned to fat. So this is Springbok analysis of muscles. You take the MRI and they basically pixelate it based on the fat. And so yellow is fat, blue is muscle, and so, or not fat. And you can see here's a case of these trapezius muscles are really, really fatty, whereas the gluteus medius muscles are actually, there's a lot of good healthy muscle here, but there is some fat in there and some pathology. Okay, this is a person. What about the pig? Well, we decided to rent an MRI. My company, Renogenics, paid for it. Tractor trailer moved on site at the pig facility, so we don't have to worry about travel and quarantine, and made a pig back. This was a pig MRI map paid for by Friends of FSH Research. Springbok Analysis has done this. You have to actually make a map of wild type pig before you can study FSHD pigs. But if you do look at an FSHD pig, what do you get? Well, if you pixelate the muscle like this here, like this biceps femoris, you can see it's very FSHD-like. It looks a lot like the glute from our FSHD patient. Okay, so we have MRI there, and again, it looks very FSHD-like. So overall, what can you do? Well, overall, you can do pretty much a really great full workup using pigs. And the benefits are that you can get relevant dosing and biodistribution. You get a very human-like immune response, human-like a transcription response. You can do your efficacy and toxicology in the pig. You can do longitudinal analyses. Now, in mice, you basically have to sacrifice a mouse to take the whole muscle out and analyze it. In humans, you do a needle biopsy. Well, in pigs, you do a needle biopsy, and you can do a before and after drug treatment. And then you can check at 3 months or at 6 months or anything. Everything you can do in a clinical trial, you can do in a pig, MRI analysis, FSHD biomarkers. One thing that's really particularly cool is you can use this to test muscle building in a dystrophic environment, right? Once you've shut down DUX4, you now got to get your muscle back and your strength back, and you can test technologies in order to get rid of the fibrosis and the fat and to get your muscle strength back, okay? So one of the interesting things, though, that we did is we did put our CRISPR inhibition into the pig. And this is, people are asking, why aren't you in clinic, Jones? Well, I'll tell you why we're not in clinic, because we did a pig experiment to de-risk our program. And what we found is that at 5e13 of our next-gen AAV, 1 week post-injection, just about 100% of the cells express Cas9. But 9 weeks, only about 25% express Cas9. And by 14 weeks, only about 5%. So what's going on is the pig immune system is recognizing the Cas9 as being bad, as being foreign, and you're triggering an immune response. This is exactly what Dongsheng Duan showed in his dog work, that Cas9, you have to worry about. This is actually a major concern for CRISPR technologies and CRISPR therapeutics in humans, in dogs, and pigs. So the pig, actually, so you might say, oh, this is a disaster. We actually look at this as great, because we would have seen this in a human, right? We would have seen this. So this saved us from going forward to the clinic, because it pointed out a problem that we had to address, which we believe we have now addressed. Okay, this data is almost a year old, and we've gone through and been able to fix this. So using the large animal model alerted us to a problem that we would have had, and I would say it saved us from having a failed clinical trial. Okay, so what's the value of a neuromuscular disease large animal model? We got durability data from the mini pig, prevented us from going forward with a clinical trial that would have failed. Okay, and some people said go forward anyway. Truthfully, some people told us that. We said, hell no, I have a conscience. Provides the opportunity to address the critical concerns in a timely and affordable way. We didn't lose time. We saved time and we saved money, because instead of 3 years from now finding out that our clinical trial failed, we're able to fix the problem before going into patients. That's how you respect the patient population. You go to clinic with the best possible cargo made, mode of delivery, and dose. We modified this system, and we believe we fixed this problem, and we're currently going through our testing of biodistribution and everything for this. In addition, the model is great for testing your therapeutic in a dystrophic situation, just like the patient population. All right, so the title was Overcoming Translational Hurdles in Musculoskeletal Gene Therapy. It created a highly effective and specific and clinically relevant CRISPR inhibition system for FSHD using highly compact, no heart and have a little heart muscle regulatory cassettes applicable to many neuromuscular diseases. Minimized epigenetic repressors that are novel. And in addition, we created an FSHD-like mini pigs for better success in the clinic. And it's a model for neuromuscular disease, fibrosis, and muscle building. All right, so we want to thank the people who did the work and the funders. Our lab, all the work I showed you was done by Takako Jones and Carice Hameda. Work funded by a number of patients that we're very grateful to, the McHitchcock Endowed Chair, and of course certain foundations that fund us, Friends of FSH Research, FSHD Global, FSHD Canada, and the Chris Carino Foundation, and NIH has provided some funding. The pigs were funded entirely by patients, Chip Wilson, the Lewis family, the Baum family, the Stollery Family Foundation, and several other families around the world have kicked in. Again, a lot of this has been driven by FSHD Canada, FSHD Global, and Friends have put in a little bit, as has Carino. Renogenics has put in a large amount for the MRI, and NIH has been funding this through the Seattle Wellstone. A lot of thanks here to Dr. Seth Friedman on help with the MRI, and Dr. Stephen Tapscott's lab for the gene expression studies and lots of advice. But the pig work that I showed you was all done by Monique Ramirez in our lab and Takako Jones. All right, the pigs are available through Exemplar Genetics. They are clones. We do have a herd coming. You can order them since November 2023. These pigs have been made available to the FSHD research community. I say freely available. You do have to pay for them, but there's no strings attached. Okay, they're not my pigs. They belong to Exemplar. And you can check out Chris Rogers, the CEO, at booth 709. Okay. So with that, I say thank you, and we'll take any questions. Oh, was not going to be time for questions. All right, so actually that went 10 minutes long. Now I did that whole thing in literally exactly, so that was 36 minutes. I did that, well, I guess I said this. But I did that whole thing in 26 minutes and 13 seconds when I was up there at the podium. So anyway, so that's the deal. It's those of you asking about our CRISPR inhibition, we've always said we are going to work out the details before we're going to take the best thing to clinic. It works. This is a concern with CRISPR approaches, and dog and primate models have shown that potentially the cured cells, the fixed cells, can be eliminated by the immune response, and that is a major concern. And we saw that exact thing in our pig, and we did not see it in the mouse. That's the key. The mouse, it looked fine. The pig, we saw it because the pig has a much more human-like immune system. And it gave us the opportunity to address this, okay, before going to clinic, okay? And I believe we have addressed it. We're still working up that data, but I believe we fixed it. If we haven't fixed it, we won't go to clinic. But I believe we have, and if that really pans out long-term, that we have good durability, well, then we're heading, we'll have a good package to go forward with. Anyway, that's our podcast for the day. Hope you all enjoyed it. Very sciencey today. And Jager, take us on home.
Thank you for listening to the MyFSHD podcast, where we share with you the latest news and information on FSHD as we strive for a cure. Do you have a question for Peter? He'd love to hear from you. You can reach him at peterjones@med.unr.edu. Thank you for listening, and join us again next time for more news and information.